Okay, with that, now it's my pleasure to introduce again Eduardo Nery, who will be talking about the second chapter of his PhD thesis. Eduardo talked to us about the first chapter last year, and he is in the final part of his studies, and he's trying to defend within the next few months. And he will be looking for postdocs. So any of you out there that have jobs available, here is job advertising. So Eduardo started working in his master's at Universidad Federal de Santa Catarina, and then after with a different group of plants, Apiaceae, Araliaceae, and after that he saw the light and came to use melastomes as models for the more broader type of questions that he is interested in about evolution, specialization, and so forth. He is now at the Universidad Federal do ABC in the outskirts of Sao Paulo, working with Anselmo Nogueira, also working with Mayara Cara, and he spent a few months here in New York as well last year. So please, Eduardo, go ahead and let's see what you got.
Okay, thank you, Fabian, for the introduction. And hi, everyone, and thank you for your time and to listen to my talk. I hope it can be useful, and I hope I can have several contributions from you all. So, as Fabian told you all, this is part of my PhD thesis that I'm going to defend around August. So feel free to comment and later on ask any question. The second chapter of my thesis I titled as Pollinator-specialized plants evolve the Selfin syndrome in the montane Atlantic forest. The work was run by me and my collaborators, Mayara Kada from NUFSC, Fabian from the New York Botanical Garden, and my supervisor, Anselmo Nogueira from UFABC. This work was conducted in the Plant-Animal Interaction Laboratory from the UFABC University, and I'm currently enrolled in the PhD program in the Evolution and Diversity program from the same university. So most of my talk is going to be about sexual reproduction in flowering plants. And we all know that sexual reproduction in flowering plants depends on pollen transfer. And this pollen transfer can occur in two different ways. Plants can exchange pollen between different individuals, which we call outcrossing, or plants can use their own pollen to fertilize their own ovules, which we call selfing. But if you look at the frequency of outcrossing across different species of flowering plants, we're going to see this pattern here, where high frequency of outcrossing is the most usual and most frequent pattern across flowering plants. And more rarely, you're going to see flowering plants that mostly reproduce by selfing over here on the left corner. And this pattern doesn't change regarding the pollination mode, either if it's abiotic or abiotic. So overall, selfing is not common in flowering plants, is rare. And why that? Most of the rarity of selfing has been assigned to reduction on genetic diversity. Here we have selfing rates for a given flowering plant and the reduction of genetic diversity in the same plant lineage. And what we see often is that in this solid line here, as selfing increases, there's a decrease in genetic diversity inside the plant lineage. Together with selfing itself, there's also a sign of background selection against the alleles, deleterious alleles that selfing let express in the phenotype. Beyond background selection, we also have bottleneck effects when these selfing-prone lineages occupy new areas or isolated areas where bottlenecks are more likely to occur, and then decreasing genetic diversity. So overall, selfing lineages are associated with reduced genetic diversity and the expression of deleterious alleles because of homozygosis, which would translate in this rarity among flowering plants. Nonetheless, we still see predominantly or mostly selfing lineages in nature, because in nature we have different conditions that might favor selfing over outcrossing. Well, outcrossing needs two conditions to occur. First, plants need a pollinator to transfer the pollen between the mates here, and plants need a mate. So when we don't have pollinators to transfer this pollen, or when we have pollinators but we lack mates to receive and to donate pollen, we have a limited conditions for outcrossing. Under these conditions that we're going to call mating limitations, selfing can ensure reproduction better than outcrossing, and it's selectively favored over outcrossing. Well, when we have these conditions selecting for selfing, most of the time you're going to see a pattern on flowering, on the flower traits. Here we have two flowers from two relative lineages. This one on the left corner is mostly outcrossing, and this one in the right corner is mostly selfing. And if we compare these two relatives, we're going to see that this mostly selfing relative usually has smaller flowers than the outcrossing relative. Also, they have a smaller distance between the anther and the stigma, which we're going to call percogamy. And usually, the selfing relative is going to have a greater pollen receipt capacity compared to the pollen transfer capacity. Here, drawn as the size of the anther, the relative size of the stigma and the anther. And these three traits here form what we call the selfing syndrome, which is a phenotypic marker for predominant selfing lineages. The Atlantic forest is this montane domain in eastern Brazil, where two interesting patterns occur along the elevation gradient. Here's a graph with the elevation gradient in the Atlantic forest, and here on the y-axis, some mating conditions. The first pattern that we know about this domain is that along the elevation gradient, the activity and availability of insect pollinators decrease. And also, we know from other studies that different plant species have reduced population sizes along this elevation gradient. So in the montane areas of the Atlantic forests, plants are more likely to face pronounced mating limitations, which would favor selfing and consequently the selfing syndrome. Hereafter, I'm gonna refer to Atlantic forest as the AF, just for convenience. And inside the Atlantic forest, we have Miconia, which is the most diverse group of woody flowering plants in this domain. And we know that from the phylogeny, the most comprehensive phylogeny of Miconia to date, that there are several radiations in the Atlantic forests that became endemic. So this endemic species provide like a natural experiment of plants experiencing this pronounced mating limitations compared to other lineages that inhabit and occupy lowland domains across the neotropics. And we also know that Miconia hold a very specialized pollination system, the buzz pollination, which relies on female buzzing bees to release pollen from the pericidal anthers. And it is expected that this great specialization regarding the pollen factor might increase the odds of pollination shorter or limitations when these insects are not available or not abundant. So the hypothesis of this study is that Myconia species have experienced an increasing mating limitation and consequently have been selected towards the Selfin syndrome in the Atlantic forest domain. To test this hypothesis has two predictions that endemic Myconia species should have evolved towards smaller flowers, smaller hercogamy, smaller pollen transfer capacity, and greater pollen receipt capacity than non-endemic species that should not face so pronounced mating limitations as those occupying the montane Atlantic forest. And also, we are expecting that this difference should have evolved under a selection-driven evolutionary scenario rather than a purely neutral evolutionary scenario. To test this hypothesis, we use the Myconia supersect discolors as a study system. Here's the overall look of the plants, except this one, my fault. And here's the phylogeny of the group. And as we can see, here in blue are those species that are endemic to the Atlantic forest. We have different radiations that became endemic. And over here with other colors, we have species that occupy other neotropical domains side of the Andes. So most of them are lowland domains. And we can see from data that we have collected in other chapters that endemic species tend to occupy montane areas when compared to non-endemic species. We collected both endemic and non-endemic species inside the supersect discolor. More specifically, 13 species from the endemic group and 38 species from the non-endemic group. We sample herbarium specimens, more specifically 151 specimens, with an average of 3 specimens per species. For each specimen, we measure 4 flower traits: flower size, stigma size, hercogamy, and anther pore size. Flower size is a proxy for pollination attraction, where larger flowers tend to attract more pollinators. While stigma size is a proxy for pollen receipt capacity, where larger stigmas tend to attract to receive more pollen. And finally, anther pore size is a proxy for pollen transfer capacity. And here, larger pored anthers tend to deplete their pollen faster and to perform fewer events of pollen transfer than small pored anthers. To infer how these flower traits have evolved in endemic and non-endemic species, we utilize phenotypic evolutionary models for phenotypic traits. We utilize different models based on Brownian motion and Ullstein-Uhlenbeck process. The BM models represent mostly evolution under neutral process, stochastic process like mutation and drift, while the OU models represent evolution under selection. We fit those models to a sample of phylogenetic trees from our study system, and we compare the fit and choose the best fit models to infer the most likely evolutionary scenario for each trait. And regarding the first trait, flower size, we found that the best fit model was the BMS model, which represents evolution under neutral process only. And this model considers just one parameter that can differ between groups, and which is sigma, which represents evolutionary rates for that given trait. And what we found is that endemic species tend to evolve slower than non-endemic species. But still, they have very similar flower sizes, which would indicate similar pollinator attraction between these two groups. And this pattern is not supporting the Selfin syndrome. And we asked why, and we hypothesized some explanations. We know from this study system that we have big differences regarding fluorescence, size, and architecture. Most non-endemic species have large scorpioid inflorescences, while endemic species tend to have smaller and glomerated inflorescences. And we know that these differences of size and architecture are related to pollinator attraction as well. And based on that, we would expect the non-endemic species to have a greater capacity to attract pollinators when compared to the endemic species. So perhaps we haven't seen a sign or a positive test of selection upon flowers because pollinator attraction might be held by inflorescence in Myconia instead of flowers. So inflorescence might be the units being selected towards reduce pollination attraction in endemic species. Our second trait was hercogamy, and the best fit model for this trait was the OUM, which represents evolution under selection. And this model assumes two parameters, one that will not vary, which is sigma, evolutionary rates, and the other one is theta, which represents the phenotypic optima for a given trait. And when we look at sigma, it does not vary between groups. But when we look at theta, we have found that endemic species have evolved towards very negative values of hercogamy, which means reverse hercogamy, when the stigma is way below the antherpore size and below the antherpore. While non-endemic species also have reverse hercogamy, but in a reduced degree. They also have reverse hercogamy, but it's not so pronounced as the endemic species. And we, well, oh, there is someone with that microphone on. So if we look at projections of reverse hercogamy compared to selfing rates, we're going to see this kind of curve here, where more negative values of hercogamy will predict high selfing rates. And what we presume or infer from the results that perhaps what you're going to see in this Myconia group is that endemic species are over here with more pronounced selfing rates, while non-endemic species also may perform selfing, but in a lesser degree. So this supports the selfing rate, the selfing syndrome hypothesis. Our third trait was antherpore size, whose best fit was the OUMVA model, which also represents evolution under selection. And this model considers three parameters that can vary between groups. The sigma parameter, which represents evolutionary rates, it showed that endemic species tend to evolve slower than non-endemic species. When we look at theta, which means the phenotypic optimum, you're going to see that endemic species have evolved towards large pored anthers when compared to non-endemic species. And finally, alpha, which is the selection strength upon that trait. We see that selection strength is slightly increased upon endemic species. Well, we know from the literature that pollen release increases as the pore size increases as well. So we can forecast a greater depletion of pollen in these large pore anthers, which would reduce the amount of pollination visitors that would actually translate in pollen transfer. So large pore anthers tend to perform fewer pollen transfer events than small-bordered anthers. So this supports the southwind syndrome in the Atlantic forest. But this trait is also related to another pattern, that is large-bordered anthers tend to release pollen even without the buzzing mechanism that is so characteristic from the buzz pollination. So we forecast a reduced dependence on buzzing bees for pollination towards the Atlantic forest. The last trait was the stigma size, and the best fit for this trait was the OUMVA model, again representing evolutionary selection, and it considers 3 parameters that can vary between groups: sigma, evolutionary rates, and again it showed that endemic species have evolved slower than non-endemic species, and theta, phenotypic optimum, which represents phenotypic optima, have shown that endemic species have evolved smaller stigmas than non-endemic species. And alpha, representing selection strength, again indicating increased selection strength upon endemic species. Well, we know from the literature that smaller stigmas can receive a reduced amount of pollen, so they have a reduced pollen receipt capacity, and this does not fit the southwind syndrome. And why that? Well, flowers are not isolated units. They work in groups, in inflorescences, which can also change the way they develop. And what we know that endemic species tend to have more congested and condensed inflorescences, and this spatial arrangement could constrain the expansion of the flower laterally, including the stigma, which would lead to smaller stigmas, not because of a selective pressure upon the stigma, but because of selective pressure upon inflorescences. And that could be just a side effect of the phenotypic evolution of inflorescences. But we still don't know that, but it would be really nice to test this hypothesis. Well, the traits that we have found here, especially large pore anthers, have been recorded in other Myconia species that inhabit montane habitats. Here by Criba and Zubado in Mesoamerica. Also, Vinny has found some species in montane areas that have large pore anthers and also in the Andes. And these large pore anthers have been associated with generalization Myconia. But there's a detail in this species with generalized pollination system. They all produce nectar. And we know that some species in Myconia can produce nectar, and most of the time the nectar are stomata in the ovaries or on the appendages of the stamens. But once nectar is produced and is available for the animals, this usually increases the diversity of animals that will visit the flower. Here, this study by Vinny, which is studying this nectar producing Myconia, we see that visitations of pollinators are mostly by nectivorous insects rather than pollinivorous or buzzing bees. So nectar production changes the pollinator community around the plant. Given that, we propose that we should analyze Myconia pollination system under a different perspective, that we can have traits that can change specificity and traits that can change dependency. Specificity is how specialized that pollination system is, where not very low specificity values would be generalization, and very specific systems would be specialization. But dependency is related on how much plants depends on those pollinators to perform opt-crossing or not, to perform a pollen transfer, not just on opt-crossing. And we suggest that nectar is this trait changing the specificity of pollination system in Myconia, because the production of nectar increases the diversity of pollinator groups visiting those flowers, while large pore anthers and reverse hercogamy are more related with dependency, because with pronounced reverse hercogamy and large pore anthers, you can transfer pollen without any kind of pollinator from the self anther to the self stigma. While these flowers over here with small pore anthers and not so pronounced reverse hercogamy, they still rely on some kind of pollinator to perform pollen transfer. Well, but we are talking about Myconia, and we cannot forget about Apomixis. Well, we are saying that mating limitations are favoring self-imprint flowers in Myconia. But we also know that Apomixis is at the corner waiting for us in Myconia. And well, Apomixis can be also favored by mating limitations because it can provide enough offspring when mates are not available. So perhaps in the system, Apomixis is lessening the selective pressure towards the Selfin syndrome. And that's why the signals that we got from the models are not so strong, because Apomixis is dealing with this pollination shortage as well. So the conclusions from this work is, first, that endemic Myconian species have evolved towards reverse hercogamy in large pore anthers, likely due to mating limitations in the Atlantic forest. Reverse hercogamy in large pore anthers are associated with a reduced pollinator dependency. We also suggest that nectar is the key trait promoting generalization in Myconia, but not reverse hercogamy in large pore anthers. And we also propose that Apomixis may lessen selective pressures upon flowers and towards selfing, because it can solve partially the problem of mating limitation. So I would like to thank my host university for my PhD scholarship, CAPES for my visiting PhD student scholarship at the New York Botanical Garden, FAPESP, which sponsors my supervisor. I would like to thank you also, my colleagues from my laboratory, and Donnie, my boyfriend, who helped me with this presentation. And that's it. I'm open to your questions.
Thank you very much, Eduardo. It was very, very interesting. Maybe I shouldn't be the person asking questions since I'm on that first slide, but I will have some questions towards the end. But anybody, now you can unmute yourselves, and if you have questions, please either put them on the chat or can ask a question. Who wants to be first?
Me.
Vini. Thank you.
Thank you, Eduardo, for your amazing talk. You are always the best. I love to see your presentations, and I like so much your work. And I'm just curious about how you consider Apomixis. Are the Apomixis also selfers, or they are in a different, in a different box of reproduction? Because I see in the literature that apomictics are also called selfers, but I think it's totally different selective pressures in the floral traits between apomictics and self-depositors. I don't know,
Selfers.
selfers that
Selfers.
deposit their own pollen in the stigma, you know? Yeah.
Yeah, there's a thing like if we see in the literature, apomixis can occur by different ways, including pollen transfer. And some
Yes, I like the
mechanisms
pseudogum.
do not rely on pollen transfer. But let's consider just the ways that do not depend on pollen. They would be also a solution for mating limitations, because if you're not receiving pollen at all, okay, I can just grow up an offspring out of the blue. So perhaps the apomictic lineages are also being selected in this pollen-limited environments. But, well, this requires another kind of data that I don't have. But it would be a really interesting question to address. And that I have asked to Ana Paula, and she told me that she had some hypothesis about.
Nice. Thank you.
Okay.
I guess Isabella or Agnes.
Isabella. Okay. And then we'll go with Agnes.
Okay. Nice talk, Eduardo. Very interesting. It's nice that you test many hypotheses that were in the literature, and then you can move forward. I have one question and one idea. First, do you think that this larger stigmatic area in endemic species are related to these reverse ergogamy? Because I was thinking about the morphology of the flower, and this reverse ergogamy would
compress.
Huh?
Would compress the stigma?
Not compress, but the selfing would be easier if the stigma is larger in this kind of architecture.
Yeah, but we found the opposite.
Ah.
So let me just comment. Let me just comment that in the dioecious species in general, which are not in the Mata Atlantica, they tend to have the largest stigmas of them all, which, you know, if it is dioecious, is by definition outcrossing. So here we see that large stigma is very well correlated with outcrossing. I mean, at least for those group in the Andes. So yeah, I see the logic, but on the other hand, who knows what's happening with that particular character? But yeah, I see your logic totally.
Yeah, we have the logic is perfect, and we were expecting that.
Okay.
I love the logic, but
I lost part of it. And the other is a question. You posit the idea that the endemic species are more limited, pollen limited, but have you tested that?
Yes, I could not test that for each terminal in the phylogeny, of course. So I had to rely on big patterns that we know about the Atlantic forests. We know that pollen limitation is always there in the Atlantic forests. We know from works from Dini and others that we have a decreased availability of insect pollinators across the elevation gradient, including buzzing bees. Yes, and we know from a few buzz pollinator systems that they tend to perform more selfing in montane areas when compared to lowland areas. So this is an assumption of our work for sure, and you can always point that, but I could not test a pollen limitation for each of the terminals for sure. Yeah.
Perhaps for a subset.
Yes, that would be a very interesting work. Yeah, I would love to do that.
Okay, thanks.
Thank you, Isabella. Thank you.
Okay, well, then I'm jumping in. Thank you so much for this great talk and for going again. This is awesome.
Every April is mine.
Nice. Well, I really enjoyed sort of the layout of obviously pollination, reproductive systems in an environmental context. That was really, really nice. And I agree with Isabella, it would be awesome if you could do experiments for a subset of these species to actually have sort of clear evidence of like what is going on. I have one question and a comment. And my comment, I'm very excited that Isabella is here too, because when you talked about nectar, you mentioned these hypanthial nectaries or stomata. I can tell you from the Andean species that I've looked at, from all neotropical tribes that I know that do produce nectar, both bee pollinated and nectar producing species have stomatal nectaries. Sorry, not nectaries, but have stomata in the hypanthium. And whenever I've checked in the field, I've never seen nectar come out of these stomata. I always see it at the bend of the filament where the stamens unfold in the process of like the flower unfolding. So I'm just, I don't know, I know obviously I've read Isabella's publication a million times and I'm always like, I don't know, I've never seen this. So this is more common question, whether you have seen anything. And then the second question, the real question is whether your Atlantic forest endemic species are all just in a certain elevation zone or whether some do spread a longer elevational gradient, because I'm wondering if some maybe do scatter more, maybe they do have some variance also across populations in the level of hercogamy or not, and so on. But yeah, here it looks pretty narrow compared to the others.
Yes.
Like I guess these are species means and not variance per species.
Yes. Overall, inside each species, they have a narrower variance than non-endemic species. So within species, they have a lower variance. So this is the second question, right?
Yeah.
And the first question is about the nectar. I could not test this in field, so I had to rely on the literature. And we know that for sure, 3 species that produce nectar in the system. 2 inside the Atlantic forest and 1 outside of Atlantic forest. So this made us think that nectar is not tightly related to pollen limitation, but instead to generalization, regardless of the pollinator environment. Otherwise, we're gonna see I guess, concentration of nectar producing species in the montane area, which we are not recording. We can see what we can see is that nectar has evolved in different times across the whole clade and perhaps across the whole Myconia genus, right? So it's a really interesting thing to look up. I guess the only one that have got closer to this question was Vinny, but it compared mostly just 1 nectar producing species in Myconia. And so we are like in a comparison with a close relative that does not produce nectar. And that would be really interesting to answer if nectar is generalized in the system or not.
Yeah, and where it is produced. This is
yes.
thing that has been studied for so long. I think even Vogel studied it like 30 years ago or 40 years ago.
Lagerheim, 1928.
Okay, that's it. Okay, thank you.
Oh, welcome.
I have a question and or a comment. I don't know, but something I keep thinking about this is you know, flower size changes, and you noted clearly that inflorescence, architecture, and size also change. And one of the things that happens is that larger flowers are grabbed by a single, like 1 flower at a time. But we've seen in the field that things that are especially glomerulate are grabbed.
Whole thing.
Yeah, the whole thing, like many flowers at a time, or maybe half the stamens from one flower and half from another flower, depending on the size of the bee. So maybe when the inflorescence is glomerulate, the flower size is a good proxy for something, but not necessarily for full attraction, because the bees are, when they're flying, they're seeing the full glomerular unit. Yeah. And I don't know off the top of my head of a good way of incorporating that in the model. Maybe having as a separate variable glomerulate versus not glomerulate, but then how do you disentangle, because they're glomerulate with small flowers and they're glomerulate with medium flowers, not so much with large flowers, but how do you disentangle the two factors? But yeah, there's definitely more than just flower size at work here.
Yes, and inside the glomerulated, we have different kinds as well. We have those that are just a stick with several flowers, and those are like different packaged flowers along the axis. So yes, I guess I was like blind by the literature, and I had tried flower size first because it was the traditional proxy for pollinator attraction. But each system has resolved its problems by different means, right? And I guess in Myconia, the pollinator attraction function is held by inflorescence rather by the flowers. So that's why we have so much variation inside this group, I guess. And I don't know an operational way to disentangle this glomerulated, scorpiated, and also because when you look very closely, even the scorpioid ones are not so clearly defined. So I guess that would be a separate moment, or even a second study addressing how inflorescence can act on pollination system in Myconia. But for sure, the secret is not in the flower by itself, as in the inflorescence, I guess.
Any other? Suzanne is commenting that the first reports on nectar are all the way back from Ule from 1895 and 1896. Yes, and in fact, some of these things he even, if I remember correctly, had some comments about pleistogamy as well in Melastomataceae back in 1895.
Seminal vascular bundles.
Huh?
In the Itatiaia Mountains, is that the
Yes,
way
yeah,
you pronounce
exactly.
it? These really
Yeah.
cool, close, funny things.
Yeah. So any other questions, comments, ideas? Just unmute yourself and go. Okay. So I do have one last question, and I know maybe this is the slide where you can explain it. So when you talk about endemic versus not endemic, how do you? Somebody could argue that if it's not endemic to the Atlantic forest, it could still be endemic to somewhere. So what is the threshold definition there, and how do you address that?
Yes, among the non-endemic, we have a few endemic species to other tropical domains, but those tropical domains are mostly lowland ones. So since the pollination limitation is more related with the elevational gradient, we can connect endemicity in the Atlantic forest to this pronounced mating limitations because of elevation. But most of non-endemic species are widespread. They are in the Amazon, Mesoamerica, and Cerrado. Of course, the pollinator environment is going to change through all those domains, of course, but it does not change regarding elevation, which is the main proxy for pollination attraction, pollination shortage, right? So, and the operational definition that we have used, it's based on the first chapter and the first paper, that endemic species have 90% of their occurrences inside the Atlantic forest. So they mostly experience the montane environment.
Yeah, just wanted the clarification for everybody in general. Thank you. Any other questions, comments? Well, thank you so much, Eduardo. That was it was great and informative. I just want to say that next month we will have Gilberto Ocampo from Aguascalientes, Mexico, talking. I'm not sure if he's going to talk about section secundiflora or about seeds or about what yet, but he will talk about something.
Yeah, I think about seeds, but I'm also not sure. He hasn't said the title yet.
Okay, okay. So that's 4 weeks from now. And then we'll have one in June. And then probably we'll break for July and August as everybody seems to be traveling, the few people we contacted and whatnot. So, and then we'll reconvene in September. Again, this is an invitation to anyone who wants to talk. We have had like one did 2 months ago, like just an idea, or it can be something more finished or something even published. So it doesn't matter. We are open to all levels, all levels of talks.
Of Dennis.
Yes. Okay. Thank you so much. And well, we'll see you soon, and we'll try to put this up on the website as soon as we can. Thank you.
Bye. Thank you all.
Bye. Eduardo,
Ciao.
don't miss the job offer. It's further up. in
There's
the
a
chat.
job offer for
There's
you.
a job
Okay,
offer.
I'm gonna grab it for
Have a
sure.
look on it.
I just opened the link.
Awesome.
Bye,
Thank you.
guys.